morning or good afternoon for those that are attending online from other places with different time zones. Welcome to the first 2014-15 Hydration Lecture Series. My name is Dr. Kavouras, and it's my pleasure to introduce Dr. Nina Stachenfeld from John B. Pierce Laboratory and Yale Medical School from the Department of Obstetrics, Gynecology, and Reproductive Science at the Yale School of Medicine and the School of Public Health. Dr. Stachenfeld is an expert in reproductive hormone effects in the cardiovascular system, temperature regulation, and fluid balance in humans. And over the last 20 years, she has developed a paradigm in humans to isolate effects of these hormones on physiological system regulation and thermoregulation and cardiovascular function and fluid balance. Dr. Stachenfeld has approximately 70 publications and has been supported through government, private, and industrial grants. And it's my pleasure to have Dr. Stachenfeld, and I did have the pleasure actually to meet her during my postdoc in Pierce, and it's a very good friend, and I'm very, very happy that she accepted the presentation. Dr. Stachenfeld.
Thank you very much. I'm happy to be here. It's my first time in Arkansas, and it was a pleasure to get a quick tour of the university, which is about 60 times as large as the university I'm at. As I was saying to Stavros and others, that real estate is so precious in the Northeast that seeing a large university is always a lot of fun. So today I'm going to talk about a number of different projects that we've done over the years to examine different aspects of fluid regulation. Most of them using similar protocols and applying the paradigm that Stavros just mentioned. So I'm going to just start by stating how long people have been examining this. If you look at this first opening statement, which I imagine that those of you who've been sitting here for a little bit might have read, you can see in 1637, this was already being researched, and it's remarkable how well Descartes understood what turned out to be very true, which is that there are nerves in the brain that understand that we need drinking when we have low body water. We sense thirst by those nerves in the brain, and we realize that we need to drink. The idea that you could figure that out with the materials that he had was pretty remarkable. So I like to mention it. I also like to mention it because usually when I share the stage with others, they begin by saying how long they've researched the topic. And I'm usually the one with the oldest quote. The title of the talk today is Integrative Environmental Physiology and Fluid Regulation. I left that broad so that because there seemed to be rather a broad interest among the different departments here. I want to start by acknowledging the people who work with me. Those people who work with me on a daily basis, especially Cheryl Leone, is my lab manager and really does an enormous amount of this work. And Drs. Mack and Nadell were my mentors when I did my postdoc, and Dr. Taylor was the one who helped me develop the paradigm that I'll discuss with you later. He's a reproductive endocrinologist and has taught me a ton about how to work with and understand the way reproductive hormones work in women. And also to some extent in men, we're just starting to do that. And also I need to acknowledge the funding from NIH and the Department of Defense, which has helped me to move forward in all of these areas. And the data that I'll be presenting today was pretty much all supported by them, and also the Gatorade Sports Science Institute and PepsiCo, which at the time I was originally funded were separate, but now are one and the same. The outline today, I'm just going to describe quickly the research program. I'll describe what I mean by body fluid distribution and control. We'll discuss a little bit about sex differences and sex hormone effects on body fluid regulation. And assuming there's time, I'll discuss effects of aging and menopause. So this is our program. We're not always working on all of these topics, but generally our primary focus has been to study the body fluid regulation. And what we've meant by that over the many years is dehydration, which is an overall loss of body fluids, as I'm sure you know. And that's a volemic hypertonic osmotic state. We've also examined hyponatremia, which is generally a loss of sodium or an excess of fluid retained, or a combination of both. Hyperosmolality, which is kind of an odd state, and it doesn't happen too much normally. It happens when we induce it in the laboratory, and that's a hyperosmotic hypervolemic state. And lately we've been examining the impact of mild dehydration on mental status. My lab has worked for many years, not so much on sex differences, but really on the impact of sex hormone exposure, either during the menstrual cycle, during hormonal contraceptive exposure. We've looked at hormonal control in both young and menopausal women, and also during hormone administration. And if you look down here, these are you can see some of the clinical issues that we've become interested in. And finally, I'll describe the KEEP study, which is really been run by Dr. Taylor, but is an important aspect to thinking about the study of menopause and hormone exposure. So beginning with body water distribution, in a normal human, you can see this is from Dr. Verbalis' paper in 2003, and you can tell from this picture that most of our body is made up of water, and it's subdivided into intravascular and extravascular space, and also the extracellular, and you can also see that a very large portion of it is inside the cells. And when we get down to this last area here, whoops, I hit the wrong thing, but you can see that we're looking here at the extravascular fluids, and this is 1/3 of the total body water. And when I'm talking about changing the level of volemia, I'm talking about only impacting the intravascular fluid, which is 1/4 of the extracellular space, or 1.3 liters, and that's a very, very tiny part of the amount of total water that we have in our body. And these two pretty much describe the same thing. I like to put them up there because different people have different ways of appreciating things visually. How is this regulated? As Descartes figured out very early on, most of it is regulated in the brain. The brain perceives changes in osmolality. It takes very, very tiny changes in osmolality before we start to see very large increases in thirst perception, and also the increase in this hormone called arginine vasopressin, or it's also known as antidiuretic hormone, or ADH. I'm going to spend quite a bit of time on this, discussing this, the way that this fluid regulation hormone works. So I'm just going to take a minute to tell you that it is, in addition to its effects on water regulation, it's also one of the most powerful vasoconstrictors in the body. So it has a very, very important effect on cardiovascular function, in addition to its independent effect on fluids. And its most important effect on fluids is the retention of free water in the kidney. So if you see an increase in vasopressin, you should expect to see an increase in body water retention generally. And since we're going to be discussing reproductive hormones, I'm going to go down to the anterior pituitary as well. And from this, these hormones are also stimulated by similar areas in the brain. Instead of working in the posterior pituitary, where the AVP comes from, these are generally mediated and released from the anterior pituitary, and stimulated also by changes that we see in the brain. The ones that I generally talk about today are follicle stimulating hormone and luteinizing hormone, and those are generally called gonadotropins. But what's important here is, in this picture, they've just called it releasing hormone, but the specific hormone that I'll discuss is gonadotropin releasing hormone, or GnRH. And our paradigm has worked by specifically blocking that hormone, and thereby changing the levels of estrogen and progesterone in the body. Just to remind you guys what happened, and interestingly, 1993 was the year that I started my postdoc, and I went up to the Pierce Laboratory to work with a very famous thermophysiologist, Ethan Nadel. And the first thing he told me was that, okay, he was interested, he was going to continue working, but he did not want to do anything related to women, and that we're going to stick with the aging grant that we had. He was not interested whatsoever in studying the impact of hormones or the kind of work that I had been doing. And I was okay with that because what I really wanted was the opportunity to work with him and later apply that to my own work. However, Dr. Nadel was greeted not too long after I arrived with some communication from the NIH, who funded all of his research. And it was basically called the NIH Revitalization Act. I'm sure you guys are fairly familiar with it, but it basically said that, especially particularly in human research, you cannot exclude women unless you have a really good reason. And a good reason by their definition was, let's say you're studying prostate cancer, you could exclude women from those studies for obvious reasons, but unless you had a really fantastic reason, there was no reason to exclude women. The usual reason up till that point was that it's harder to study women, and because we had to understand changes across the menstrual cycle of hormone exposure and so on and so forth. So he had to change his attitude, and they told him if he didn't start including women in one of his ongoing studies, which was a study we called the blood volume study, I'm not even sure the name of it, and include women in the study that we were just beginning with, which was on aging, that they would pull his funding. So he became suddenly interested in studying women, and they gave him the opportunity to sort of make up for lost time. So we in all of our studies from then on, we did start to include women, and that made things a lot more interesting for me. For some reason, got no slide here. Okay. Why do men not want to study women? Well, this is generally why. Because, I shouldn't say men, that's not fair. Why do people, scientists, not want to study women? Part of it had to do with the fact that there's large fluctuations of these hormones across the menstrual cycle. On top, I'm showing you estrogen and progesterone, and you can see estradiol changes across the menstrual cycle. It has two peaks, one large and one small. This peak occurs right before ovulation, and progesterone has a small change, but really doesn't have a very large effective peak until the end of the menstrual cycle, which is generally called the mid-luteal phase. And down here you can see related changes in the gonadotropins. But women had specific risk factors for dehydration, and some of them had to do with different responses to environmental conditions like temperature and even temperature perception, some people think now. There was discussion about drinking habits, longer running times, and aging, but one of the things that we had not discussed very much was the impact of sex hormone exposure in women and how that might impact their susceptibility to dehydration. So I'm going to just introduce you to this protocol. There's one that's very similar to it that I'll show you in a second, but we use this a lot to study osmotic regulation of arginine vasopressin, and also to study osmotic regulation of thirst. And it's a pretty simple protocol. We had, for the dehydration studies, we used exercise in the heat. The subjects came in and exercised for 2 hours. And for these studies, we were using women because we were specifically testing the effects of female reproductive hormones. So we would have the women come in, they'd exercise for 2 hours. They would tend to lose between 2.5% and 3% of their body weight. There would be a short break, and then we would let them rehydrate. We began with controlled drinking and also used ad libitum drinking, but we found out that they turned out to be very similar. So we went back and forth with that. But generally, when we were really trying to study what happened with regard to thirst, we would use ad libitum drinking to use drinking behavior as a surrogate for thirst. Now, as I mentioned to you before I leave here, the dehydration would cause a hyperosmotic hypovolemia. We did exactly the same protocol, but this time we used hypertonic saline infusion. Sorry about this error here. This should say an infusion of 3% saline. And when we do that, we draw a lot of fluid out of the cells because of the hypertonic environment. That creates a very, very large plasma volume expansion on the order of 15 to 20%. So it's extremely a large stimulus. And with the dehydration studies, we would tend to get, the lowest we would tend to get would be 7.5% to 8% loss in plasma volume. So if you compare the two, the hypertonic saline infusion should be a mixed message to the brain in a sense, because it would be receiving message that in a sense it's overhydrated, but at the same time it would be receiving a message that osmolality is very high, and it should start to stimulate things like AVP and responses like AVP release and thirst and drinking, because of the high level of osmoles, or in this case sodium. Dehydration, it should be receiving similar messages saying that it's basically the body's dehydrated, we're thirsty, we want to retain water. Just to begin with sex differences, this is what would happen. We would see, you'll notice here, pretty much all of these graphs are going to have osmolality on the x-axis and either thirst or AVP on the y. And these are the graphs that we describe as we use to describe osmotic regulation of thirst, or if AVP is up on the graph. And you can see something interesting that happened that was very interesting to me right away. We were looking at sex differences, and we noticed that the men are in green, and women in the follicular phase are in red. And you can see there's absolutely no sex differences there. But because there's a difference in osmotic regulation of thirst within women across the cycle, you can see that there was a very dramatic difference in osmotic regulation of thirst between men and women at this point in the cycle. And one of the reasons I always found this to be particularly important is because when we describe sex differences and when people measure sex differences in the literature, I always see them comparing men and women in the follicular phase. And in the follicular phase, if you'll remember, that's when all the hormones, particularly estrogen and progesterone, are at their absolute lowest. So when you examine women and men together when women's hormone exposure is at its absolute lowest, you don't see much of a sex difference. You do see a sex difference when hormones are elevated. And this is, I put this up here. I don't have data on men with plasma osmolality and vasopressin, but you can see the clear difference across the menstrual phase. And when we looked at this again with saline infusion, you can see very well here that the men that we basically had the same response with the release of arginine vasopressin. So this pretty much matched what we saw in the thirst. That the big differences between the men and the women occurred when the hormone levels were at their lowest. Actually, with AVP, there were differences in both times in the cycle. So you could easily argue that there are important sex differences between men and women during saline infusion, but not so much during dehydration. One of the things also that we found that was different between the sexes was the response. During saline infusion, the men held on to a lot more of their sodium. We were pumping quite a bit of sodium into them, and the men retained a lot more sodium than the women did. Interesting, there wasn't much of a dramatic or important difference in body fluid balance, and we've never really been able to figure out exactly why that's the case. And I apologize for missing the legend here, but one of the most important aspects of those protocols was that to go along with the sodium retention, we also saw a nice, robust increase in blood pressure when we infused sodium into the men, and no change at all in the women. So just to summarize that, it's important when you study sex differences to control for changes across the menstrual cycle. I want to remind everybody here that the early follicular phase is the first 6 days of the follicular phase typically in women. That's because around 7 to 8 days we start to see that rise in estrogen. And you want to remember that given the fact that the menstrual cycle is 28 days, if you look at this across the lifespan of a woman, you have to remember that that's a very small percentage of her reproductive life. So if we're going to describe sex differences across men and women, and we're going to say, well, I examined this in the follicular phase and I didn't see any difference, you have to always remember, if you're reading or if you're thinking about designing your own protocols, that that really isn't necessarily an accurate interpretation. That doesn't mean that it's wrong, but we really have to remember to consider different phases of the cycle. For that reason, I never recommend that we automatically combine men and women into the same group. And you do, if you can, want to match on all the characteristics between the men and the women as much as possible, which is, I don't have to tell you guys how hard that is when you think of all the different ways that men and women are different. And I'm going to move into talking about fertility, drugs to control hormone levels. So I'm just going to touch on hormonal contraceptives. Hormonal contraceptives we've used as an intervention, but now I see an awful lot of studies where women are accepted into studies whether or not they're on oral contraceptives. And you want to remind yourself that taking an oral contraceptive exposes women to a massively high level of hormones, much, much higher than endogenous levels. So you have to be careful about the type of pills that they use. And if you really want to be careful, nobody on oral contraceptives should be included in studies. Or, and it's simple to do that. You can ask women to go off for a month, and there's different ways of dealing with that. But if you do that, it really compromises your ability to interpret the data. So I'm just going to remind you again, this is what the normal cycle looks like. And this is what the cycle looks like once we give hormonal contraceptives. Typically, there's a placebo period here, which is during menstruation. The women start to take the pills. Hormone levels go up extremely high. And sometimes we take the combined pills. And sometimes for our studies, we've given hormonal contraceptives where we give estrogen first, and then we add progesterone. This is a study design. If you're just examining women who are taking hormonal contraceptives, both hormones are likely elevated. And also, you should remember that when you give oral contraceptives, the endogenous hormones are suppressed. So if you try to measure hormones in the blood, they're all going to be very low, but that doesn't mean that the exposure is low. So these are studies examining reproductive hormone effects on water regulation within women in studies where we used a combined oral contraceptives. And one of the interesting things that comes out right away, this is osmotic regulation of vasopressin. You can see that there's a very large shift. This is the follicular phase in blue, and the progestin-only contraceptive in red. And you can see that there's a very strong shift to an earlier release of arginine vasopressin during the luteal phase when endogenous estradiol and progesterone are elevated, and also when we include estradiol in the progesterone pill. And this is similar here with thirst, just not nearly as dramatic. And I've always sort of accounted that to be seeing, these are thirst ratings, so they're a little bit more subjective than measuring a hormone in the blood. But it's important to note that progestin is a little less effective, at least with regard to osmotic regulation of ABP, when there is no estradiol around, and that might be due to effects of or lack of effects on estradiol on progesterone receptors. And this is a table that pretty much tells us the same story. These are the x-intercepts of the two, and what these tell us is that if it's a lower number, it tells us that it's shifted over, and it really would require less of a osmotic stimulus to increase ABP. Okay, so getting back again, using fertility drugs to control hormones in young women. So this is one of the ways that we've done this, and we're actually involved in doing this now. One of the reasons we're doing this is not because we really want to change the hormones as much, but because we are mostly interested in the impact of these specific kinds of contraception. The more interesting way that we've done this is by using gonadotropin-releasing hormone antagonists or agonists. Leuprolide is acetate is a very common drug. It's called Lupron, and if you know anybody who's had prostate cancer, it's also used in men. And what it does is it does what it sounds like. It's an agonist for the GnRH, that hormone that I showed you before in the brain. And initially, it causes a very large increase in GnRH, and that's subsequently suppressed. And then after that, you see, after this surge in estradiol, you see a rapid fall, and then this will stay down for probably about 6 weeks before it starts to wander back up. We don't do our studies that long, but the problem with this particular drug, it's an agonist, and it takes about 3 weeks really for that drug, for us to be certain that that drug has really suppressed estradiol and progesterone. And then we can do our studies here, and we can add progesterone if we want to. We've also used this, we've started to use these types of drugs with testosterone as well in certain populations. Luckily, an antagonist showed up on the market, ganirelix acetate, and that's a really nice drug. It's an antagonist. It works much faster. It suppresses GnRH and estradiol very quickly, within about 2 days. So if you notice here, this took us 70 days to complete our studies. Now it takes us about 40. So it's really nice for us, and also really nice for the subjects. But what this really enables us to do in a laboratory is enable us to start to isolate the effects of whatever parameter we're interested in. And just to show you a little picture about how that works. This is where that drug acts. It competes with GnRH for the GnRH receptor in the arcuate nucleus and suppresses gonadotropins and ultimately suppresses all ovarian production of estradiol and progesterone. Show you what happens when we do that. This is, I'd like to put this slide up because just to, in the interest of full disclosure, because this is the worst one that ever happened to us. When we have the GnRH antagonist, you can see very clearly that it very much suppresses estrogen and progesterone. When we give estrogen back, you can see that it increases estrogen, but not progesterone. When we give them both back, you can see that both hormones increase, estradiol and progesterone. What was weird about this study, and it never happened again, was that when we gave the two hormones back together, we had a much larger response in estradiol than we did with estradiol alone. I thought that was going to continue happening, but it didn't. Generally, estradiol goes up and pretty much stays at the same high level. So that allows us to examine our parameters of interest with a nice model that allows us to control estrogen and progesterone. And actually, this can be applied to any study that you're interested in doing. It doesn't have to be on fluid regulation. It could be on any, any particular area of interest. Right now, in fact, rather than studying fluid regulation, we've been using this paradigm to study cardiovascular effects and endothelial function in women with polycystic ovary syndrome. And instead of using estradiol and progesterone, we're using combinations of estradiol and testosterone. We're using testosterone alone, and so on and so forth. I can imagine that you can see the possibilities here. As long as you have a qualified reproductive endocrinologist, which I was lucky enough to find in Dr. Taylor, who can monitor the subjects, it really does create to me, it just opens a world of possibilities. And what one of my colleagues, I always hated when he said this, but he called it medically oophorizing women. And it's an ugly thing to say, but in a way that is what we're doing. And it allows us to have the same kind of freedom and interest to do in human studies what people have been doing in animal studies for years. So that opened up a world of possibilities to us, and we've been working with that paradigm ever since. Just to close this particular area, I don't know if you guys have been following this too much, but the same thing that happened to Dr. Nadel in 1993 has happened to a lot more scientists now who have gotten the edict that NIH plans to insist on balance of the sex and cell and animal studies. So with the animal studies, it's required or will be required soon that female animals are included in all of animal studies unless there's a good reason not to. And the same is true for cell studies as well, even cells in culture. And there's a big hullabaloo about that. A lot of people think that that's completely unnecessary, but the point is that there have been a number of times in science, and particularly in neuroscience, where there have been discoveries in which cells that have been generated from either from male animals or male sources that respond differently than female sources. So the discussion is now that this has to be rectified. And it's being rectified to the point where I saw an announcement that I think there's an $8.2 billion fund being established for investigators to go back, and they can apply for this fund and go back and redo some of their studies in females, which kind of makes those of us who's been studying females all these years a little bit mad, because the NIH dollars are highly sought after, and any time that you put money like 10 point anything billion towards something, you're not getting it somewhere else. So that means other studies that are related to women's health will not be funded, so that these laboratories can do what they should have been doing all along. But that's just, I had to get that off my chest. Okay, I'm just going to move on to hydration and aging. And I understand that Dr. Kenny was here, and I think that he might have described some of these data. So I apologize if I am redundant with here, but he was here last year, correct? So maybe some of you guys, this will be new to. When I first came to the Pierce, they were studying, the grant that Dr. Nadel had just received was to study body fluid regulation, thermoregulation in aging. And this was the first study they did. This was before I got there. They exercised older men only, and they measured changes in their thirst ratings as they did it, and also their fluid intake. And as had been demonstrated before, older subjects had a lower or attenuated thirst response to dehydration. But one of the interesting things, and this study was done by Gary Mack, and the interesting thing that Dr. Mack noticed when he did this was he would not describe older people as dehydrated, which is what a lot of people describe them as. He would argue, and did argue, and I think was correct, is that they have moved a little bit. They've shifted on the osmotic curve. So if you look over here, and that's what this graph here is showing, you have the younger people, they exercise, they become dehydrated, they're permitted to drink, they come back to where they started. Older people, they're a little hyperosmotic when they begin, they exercise, they increase, not quite as much as the younger people, but they increase quite a bit. They start drinking, and they come back exactly to where they started. If they were dehydrated and were drinking the correct amount, they should have come back to what we would have considered normal, where the younger people are. But indeed, they didn't. And we thought a lot about that and why that would be. One of the theories I always had was perhaps it's somewhat of a protective effect. Older people have a stiffer vascular system. Maybe it's better to have a little less water around. Their kidneys don't work as well. so they aren't putting as much strain on their kidneys by taking in less water. We don't really know those were theories, but it seemed to me to be a perfectly reasonable thing that older people would not be necessarily dehydrated, but would be operating on a different part of the curve. And just so you know, when we examined this during hypertonic saline infusion, and this included both males and females, by the way, we found that there was absolutely no effect during hypertonic saline infusion on vasopressin or thirst between young and older people. And we were speculating as to why this would be, but it was very clear that especially thirst was attenuated with age during dehydration and not with hypertonic saline infusion. And to make this more clear to us, we actually, since we were together in the same lab, we had access to all of the data, and we combined the data from the two different studies. So this is just thirst. We didn't have data on AVP in the first study. But when we combined osmotic regulation of thirst in the younger people under the conditions of hypertonic saline and dehydration, and older people under those same two conditions, you can see that under dehydration, which is in these open circles, there was a much steeper incline in that relationship compared to hypertonic saline infusion. In the older people, you could see that there really wasn't any effect, that it didn't matter whether their brain was seeing hypertonic conditions under dehydrated conditions or due to osmotic infusion of AVP. And so if you remember, the difference between these two conditions is volume status. So our theory became, and this I just drew this out to make it a little bit more obvious, but our theory became that it was due to an inability that the older people had less of an ability to sense changes in volume. Now, it's very hard to isolate volume from osmotic stimuli during fluid changes, because if you do anything, such as dehydration, you're going to be changing them both. Except we had help from a guy named Arthur Du Bois, who was one of the leaders, actually a leading pulmonologist in his day, but he had experience with this technique of head-out water immersion, mostly because his dad was even more famous than him and did this kind of work. And they were both at the Pierce lab. They were not both at the Pierce lab, but the younger guy, who at the time was, I think, in his 70s, helped us set up a water immersion tank. And why did we want to do that? If you do this, if you put people in a water tank, you will restore. If you put dehydrated people in a tank of water, you will restore their volume, but you will not change their plasma osmolality. And the reason for that is that the restoration is not because you've given any fluid; you're just moving fluid around due to hydrostatic pressure. And that's basically what we did with this. We had people, older and younger people, dehydrate at night. Then they slept in the hospital and came back the next day, and they were still dehydrated. They were still down by about 5% or so in their plasma volume. We put them in a tank of water, and also they had a time control where they sat in a water-perfused suit. And we measured them during control. We put them either in the water tank or in the suit, and we let them just sit for a little bit, and then we let them drink water. And this is what happened. If you look up here, these are the younger people, and if you look down here, these are the older people. And you can see that as soon as we put the younger people into the tank, and those are the dark circles right there, their thirst started to drop immediately relative to their responses in the water-perfused suit. This, on the other hand, had no impact on the older people. So that supported our theory that that the older people were less sensitive to volume changes. This did as well when we let them drink ad libitum. You can see when the younger people who were quite dehydrated, they drank quite a bit when they were sitting in the control or the water-perfused suit. And their interest in drinking was very, very much suppressed when they were in the water tank because their volume had been restored. But this did not happen with the older people. These were some of it was statistically different, but relative to what we saw with the younger people, it was certainly minimized. So we theorized then that the thirst deficit in the older people was a function of attenuated volume, sensitivity, or perhaps baroreceptor function, and there is evidence in the literature for altered baroreceptor function in older people. Shifting to an important topic nowadays, I'm going to stay with old people, but I'm going to move to menopause. And if you look what happens in menopause, this is premenopause, and this is what is going on with the hormones. It's a sort of a squished view of what I've been showing you spread out. And you can see that there's a great deal of regularity. If you look here in perimenopause, things start to get a little messed up. And it's what happens is with estradiol, for example, is it still is fluctuating during perimenopause. And this is why the ability of physicians to let a woman know if she's in perimenopause or not is so difficult because, or it is not typically done by taking a blood sample, because if she happens to go to the doctor this day, she's going to say, oh, your estradiol is fine. You're not perimenopausal. She goes here, she might say something like, oh, well, you know what, you might be already menopausal. So they typically let women know that they're in perimenopause with their symptoms, when they start to see symptoms or irregular menstrual cycles, and they wait a year after the final menstrual cycle to describe a woman as menopausal. And here you can see the more familiar view of postmenopause, which is low estrogen and progesterone, and fairly elevated levels of the gonadotropins because of the lack of feedback coming from the ovarian hormones. This is what starts to happen. I'm not going to go through everything because first of all, it's too awful, and second of all, it's also that I don't study most of it. What I have been interested in is cardiovascular disease, and as you can imagine, with all of the work that we've done using those paradigms, we've also examined the difference, what happens with estradiol exposure in postmenopausal women and things that affect fluid regulation. And here's just one more graph like that. This is a graph of osmotic regulation of AVP. In postmenopausal women, these people are way past menopause. They're not taking estrogen. We put them on estrogen, and you can see that estrogen, this is baseline, but estrogen shifts that curve so that there is an earlier release of AVP at any given level of osmolality. And that's sort of similar to, and makes them in a sense similar with regard to comparing them with young women. So this isn't necessarily a good thing. It's just that we were very interested in examining estrogen effects on osmotic regulation of AVP, and older women are an easy model to use because they have very little estrogen exposure on their own. One of the interesting things to us also is that we found that when in direct contrast to changes in estrogen exposure and osmotic regulation of AVP in young women, which we didn't see, if you recall, we didn't see much of an effect on fluid balance. In the older women, we did. We saw a reduction in, with estrogen, we saw a reduction in urine output, and an increase in overall fluid balance relative to control when they were not taking estrogen. So in comparison, really, if I were going to guess about an aging effect, that estradiol might have an important aging effect, if you compare older and younger women, in the fact that giving older women estrogen causes them to retain a little bit more fluid, and it doesn't have that effect on younger women. This is an interesting topic as far as giving older women estrogen nowadays. It became quite controversial. I'm sure you guys all heard about the Women's Health Initiative, where it was suggested that women stop taking estrogen, or that's how that was interpreted. But it was suggested that there might be increased cardiovascular risks in women taking estrogen. Then that study got hammered, and the reason that it got hammered was it did something very strange that you would never do clinically. It gave women estrogen when they were already 60 to 65, and that's never done. It's never done because there are already changes in the cardiovascular system that make giving estrogen a risk. So a physician, if he or she is good, is not going to say, you're 65, I'm going to give you estrogen. First of all, there's very little reason at that point. It doesn't necessarily improve cardiovascular disease as they demonstrated in the WHI, but also at the time that women most need it, which is treating the symptoms of perimenopause, has long passed. But in this study, they did that. And the press got a hold of it, and it really wasn't totally the author's fault. But the press got a hold of it and twisted things around a little bit, and it made things seem as if all women taking estrogen were at dire risk of developing clots, strokes, and other types of vascular disease. They also did not show much of a benefit in these women on cardiovascular disease. So this began this Kronos Institute. They began to support sort of a study to go back and kind of redo so much of the data that had already been out there in a systematic way in a group that they believed more appropriate to study this in, particularly younger women. And what they call this the KEEPS trial, the Kronos Early Estrogen Prevention Study. So it was called the KEEPS trial. This was done all over the country. It was a 4-year double-blinded placebo-controlled clinical trial, so it was beautifully designed. And they used low-dose, either oral, transdermal, specifically a skin patch and a sickly monthly progesterone. The women were healthy, ages 42 to 59, with a mean age of 52. And this is just about the time clinically women are interested in starting to take estrogen therapy. And they began with an N of a little over 700. And the trial excluded any women with cardiovascular disease risk and all of the other risks that are generally associated with taking estradiol. Okay, so they used a number of different centers across the United States. The Yale OBGYN department was one of the centers, and the guy who's now our department chair was the PI. This was basically what the study was. They gave a fairly normal amount, or a standard, a clinically standard amount of estradiol and progesterone. And this was lower than what was given in the WHI. And just to cut to the chase, 64% of the women, which is pretty good, stayed in the study. They completed all 4 years, and there were a bunch of women who discontinued but were followed throughout the study. Some women dropped out when they figured out they were on the placebo and they really did want to be on estrogen. And they found that hormone therapy, not to anyone's surprise, reduced the symptoms of menopause, especially hot flashes and night sweats. They found some improvement in bone density. and generally improved sexual function compared to the placebo group. They had similar rates of breast cancer, endometrial cancer, myocardial infarction, TIA, and stroke. And there was no change, especially important with regard to comparing it to the WHI. They did not see changes in any of the clotting outcomes. And the main difference between this study and the WHI was that they were younger, healthy subjects. And the reason I know the reason I let you know that some of this was done at Yale is because you obviously would consider me biased, and I will fully admit to that. But one of the things that makes this study powerful is that it really was done on women who would, at a time in their life when they actually would be taking estradiol. They had no effects on progression of arterial wall thickness in the 3 treatment groups. So that tells us also that even though the estradiol didn't do any harm, it didn't necessarily protect these women from cardiovascular disease. And a primary marker, coronary artery calcium, showed a trend for less progression in the 2 hormone therapy groups, but again, that wasn't excitingly powerful. So I'll stop here and take any questions if you have any. Thank you.
Thank you, Dr. Stachenfeld. Any questions?
Thank you. Great talk. I had a question about the return to hydration status after dehydration with older and younger individuals. With the dehydration, you did a small amount of exercise, right?
Right.
To induce the dehydration. Was there any implication that that exercise intensity could play a role in the stimulus of water consumption afterwards, or is that something that is kind of taken into account because of their relative exercise intensity?
Well, I think that's a great question in general, because they did do, well, it wasn't a little exercise. They did 2 hours, and they both groups lost about 2.5%. The older group lost a little bit less. They did exercise at a lower intensity, but at a similar relative intensity. But that's a great question. It always has bothered me because they are indeed exercising at a lower intensity. It always would be nice if we could have a third group, or even have the younger group come back and exercise at that older people, just to make those kinds of comparisons. I don't actually think that the exercise itself had an impact on those specific findings, basically because it's been quite reproducible. But that's a great question. I don't know that you can really answer it. In a sense, you'd have to kind of do the same thing you'd have to dehydrate them again without exercise, perhaps heating, and then you've introduced, heating. But if you could, sweat people and check the same thing, I think you could find it out that way.
Any more questions?
No one has any questions?
I have a question for you. One of the thoughts that I have, I was looking at people that what they've been described in the literature as low drinkers, people that they are just naturally do not drink much water. So people that they are like total fluid intake, I would say 1 liter or less per day. And then we have people that they are what has been described as the high drinkers. So if you compare those 2 groups, do you think there is any difference in their osmotic regulation of thirst and ABP just because they've been they've been adapted? I don't know if the word adapted is correct. Do people adapt in the low and the high drinking? Do you think that the osmotic regulation will be different?
So are you asking whether that would be something that's like genetic or something that is an adaptation to
as an adaptation,
availability
or if you take
of water?
somebody who doesn't drink, let's say you get a population that there are people that they develop kidney stones, for example, and their doctor says, now you have to start drinking more water if you don't want to come back here. So when these people are moving from low drinkers to high drinkers, and they increase their water intake by 1 liter or 2 per day, does something change in the osmotic regulation of vasopressin? Do you have any? I mean, I know there are no data in the literature.
There are no data for that. One of the things that I really believe is that humans are generally low drinkers. So their regulation, their ability to sense low volume in their body is very poor. And the people who drink excessively or drink, more than or drink appropriately, do it because they've been told that it's good to drink. Like that person, you're talking about an extreme person, but even, I remember, I don't know if it's still the craze, but you would go to New York and there were women, dragging around like huge bottles of water because they thought that, for some reason people started to believe that drinking water was healthy. I remember when we were working with some of the older women, they told us that their mothers told them to drink it because it was good for their skin. So I actually think that people, their thirst sensation in humans is poorly developed, and people naturally are low drinkers. And they only become, high drinkers when they're told to be. And, that's one of the impetus behind some of the studies that we're pursuing with regard to mental status. So if you have a person that's a low drinker, which is most of us, they don't drink all day long, does this impact their cognitive function by the end of the day when in a sense they're a little bit, they're less hydrated than they were. And we know from our personal experience, if we don't tell our subjects specifically to hydrate, they all come in dehydrated, or I would say with, very high urine osmolality.
Anybody else? Can we use the microphone just for the online viewers?
Have you looked at women with altered hormonal status, like individuals with PCOS, in these dehydration and rehydration studies, and looked at the effects on that?
Yeah, we did a study on where we induced dehydration in women with PCOS on and off testosterone. And one of the things that, first of all, it's very tough to get them to dehydrate. But one of the things that we noticed was that they had very similar responses when we had them suppressed with a GnRH antagonist. But when we gave them testosterone and when we gave them estrogen in both conditions, they were much more sensitive to the changes in those hormones than were the control women. So we gave they had an earlier threshold for thirst and AVP release, more so than when we gave those hormones to the control women.
Thank you very much. I really appreciate it for your presentation.
Thank you, guys.
Appreciate it.
Hi, I'm JD Adams. I'm working on my PhD in exercise science here at the University of Arkansas. Exercise science, simply put, is understanding how to maximize the body's health and performance. I'm interested in the science of heat and hydration in both healthy people and people with medical conditions. Let me show you what students and faculty are studying in the Human Performance Laboratory at the U of A. Concussions are a serious concern for athletes of all ages. Dr. R.J. Elbin studies the neurocognitive, physical, and psychosocial effects of sport-related concussion in youth and college-age athletes. His research focuses on identifying factors that influence concussion risk and recovery. Whoa! Dr. Michelle Gray does research on functional fitness to help older adults live independently as long as possible. She studies muscular power, an aspect of fitness that gets to the heart of the matter. Maintaining adequate muscular power helps prevent fall injuries, the leading cause of both fatal and non-fatal injuries among older adults. Well, how'd I do?
JD, I've got good news and bad news. The good news is you survived the test. The bad news is the worst reading we've ever seen.
Man, I need to work on my core strength. Dr. Barry Brown created this device called the AB test to quantify core strength. His research has shown that sit-ups measure abdominal muscle endurance, not strength. Building the core reduces lower back pain and enhances athletic performance. Wow, there's some fascinating stuff in here. Diabetes begins with insulin resistance in the skeletal muscle. Dr. Nicholas Green is researching how we can use exercise to combat those effects. His research is working toward ways we can prevent diabetes and related diseases or lessen their severity. Dr. Tyrone Washington's research focuses on how different physiological conditions, such as obesity and aging, affect muscles' ability to recover from damage. Nothing like water to quench your thirst, but too little water can have a big impact on a person's health, exercise performance, and cognitive function. By artificially inducing thirst and monitoring the body's response, Dr. Stavros Kavouras shows that even small degrees of dehydration can also affect kidney function, glucose metabolism, and hormonal changes. Man, this suit is hot, but researchers use it to study body temperature control. Dr. Matthew Ganio's research focus is on understanding how hydration and heat stress affect people with medical conditions such as diabetes and obesity. The findings from this research have implications for how to improve long-term cardiovascular health. Man, it's hot in there, but Dr. Brendan McDermott needs a heat chamber like this to do research on prevention and treatment of exertional heat stroke and hydration status in athletes. His studies can have profound benefits not only for athletes, but also people such as soldiers and those who work in extreme weather conditions. Well, what do you think? Pretty cool stuff, huh? Many of our graduates go on to work in clinical, sport, and academic settings. If any of this interests you, visit our website. You can be a part of the important research going on to help people live healthier lives. We look forward to hearing from you.